I want to go ahead and introduce our next presenter, Monica Embers, who also happens to be from Kansas, which is near me. I'm from Kansas City, Missouri, but she's from a smaller town outside of Wichita, Kansas, and we both laugh that growing up in that Midwest part, you don't have to only worry about ticks, but there's these things called chiggers that we don't even, if you're from the Midwest, you know what I'm talking about, right? Nobody knows what that is here when I say chiggers, but anyone from the Midwest knows that if you wanted to play outside, that was just what happened to you. You got bit up. So Monica Embers obtained her Ph.D. in the Department of Microbiology and Immunology at the Pennsylvania State University College of Medicine, where she studied immune responses to papillomaviruses. She made the transition to study of bacterial pathogenesis when performing her postdoctoral research on the Lyme disease spirochete at the Tulane National Primate Research Center. She also has joined the faculty there at the Tulane National Primate Research Center. So I'm going to welcome up Dr. Embers, and I know you're really going to enjoy her presentation too. Each one of these presenters are terrific, so thank you.
Well, thank you for having me. I appreciate being here, and I sometimes tell people, my colleagues in the scientific field, that I'm privileged as a Lyme disease researcher because I get to meet the people who are affected by the research that I do. And most people, you know, if they study different diseases, they're kind of stuck in the lab, and there isn't a lot of outreach. And so it's really an honor to be able to meet patients and hear their stories, and it's also very motivating for me. So I'm going to talk to you today about the immune system and how important it is in this disease. And I'm going to talk to you about the challenges that we face in trying to the challenges that we need to overcome in order to diagnose and treat this disease. And at the end, I'm going to talk to you about some of the things that we're doing. So this is just a brief overview. I'm just going to briefly cover diagnostic testing. I'm going to talk about how the spirochetes avoid immune recognition. They're very sneaky. And how the spirochetes and the ticks actually suppress the immune response. And how our immune response affects both diagnosis and treatment of Lyme disease. And then I'll talk about a new project that we have underway too. So the CDC case definition for Lyme disease is that you must have either an EM rash or at least one late manifestation laboratory confirmation. So as Mary Beth talked about, the two-tier test, that's the laboratory confirmation. And or you can have the isolation of Borrelia from a skin biopsy or blood, and that's very rare. That doesn't work very nicely. So what you're looking at here is each line is the antibodies to a particular antigen. So we look at 5 different antigens at the same time. And you can see here that we have 5 monkeys that were untreated and 5 monkeys that were treated. And this monkey didn't seroconvert at all. There were no antibodies that we could detect. But guess what? He was infected. I found spirochetes in his heart 12 months later, and he didn't have a second exposure. This monkey generated responses to 2 of the antigens, but they were all gone by 12 weeks. So if we're trying to diagnose this patient, we're missing it completely. So this just proves that seronegativity can happen. And what controls that, we don't really know. So this just talks about diagnostic testing and what's needed in order to establish, considerations. But the point I want to make is that the accepted tests for Lyme diagnosis rely on a very strong and polyspecific antibody response, which doesn't necessarily happen. So We know that Borrelia persists in reservoir hosts. So in nature, mice are reservoir hosts, and they fail to clear the infection, but that's how it needs to be, because the spirochete wants to survive in the reservoir host so it can propagate to the next host. And so the fact that mice don't get very sick or they don't clear the infection makes sense evolutionarily. So the spirochete has all these different tactics it uses to prevent immune recognition. And we think that those tactics that they use also come into play when an incidental host is infected, and we are incidental hosts, and our non-human primate model are incidental hosts. And right here, if you see this little red line right here, it's kind of hard to see. This is a heart of that monkey that was seronegative, and this is a nice, pretty spirochete right there in the middle. So we know that Borrelia evades the host immune response in a lot of different ways, and there are two main ways. One is that they can actively suppress the immune response. So they affect host cells so that they don't make the kind of responses that they should. And then other ways is that they hide, and they can hide in different sites. They can hide from, antibodies and blood proteins, a lot of different things that these spirochetes do to avoid being recognized. And I'm not going to talk about that. And the other thing is that the ticks suppress the immune response. So when the tick feeds, it's effectively regurgitating its contents and its saliva into the skin and the bloodstream for several days. And that tick saliva has very potent immunosuppressive properties. So when we think about generating vaccines or against Lyme or other tick-borne diseases, we have to overcome that immunosuppression that is imparted by the tick as well. And we also don't know how long the immune suppression lasts, because that saliva could linger in the host for quite some time. So when it comes to treatment, Mary Beth talked about our study in non-human primates, and it makes sense to me because doxycycline is an antibiotic that is microbiostatic. That means it doesn't kill the bacteria. It relies on a good immune response to clear the infection. And these spirochetes are evading the host immune response. So to make a long story short, what we found, I thought that patients who had lingering chronic symptoms, had a lingering infection, I thought they would have a persistent infection and they would have continuous antibody responses. But that's not what we found. What we found is that patients who generate good antibody responses to begin with are the ones who return to health. And so these are B cells, these are the antibody producing cells that are taken from patients at the time that they're diagnosed. And this was a collaboration at Stanford, and what they found is that patients who return to health have really lots of good antibody secreting cells to begin with. And we did the same thing with our test. And we looked at, I don't know why this title's gone. We looked at their response to those 5 different antigens, just like we did with the monkeys. And the patients who ended up healthy generated good antibody responses to all the proteins, or a lot of the proteins. And the patients who ended up developing post-treatment Lyme disease syndrome did not have very good quality antibody responses. So that's just stated here. So we want to try to understand better how the Borrelia are able to suppress the immune response in incidental hosts. If you were here yesterday, Nicole Baumgarth talked about a study that she did where she infected mice with Borrelia, and then she immunized them with a flu vaccine. And she found out that the mice that were infected with Borrelia didn't generate antibody titers to flu. So what is this pathogen doing to suppress the immune response to something completely unrelated? So that, that's reservoir host, that's a mouse, and maybe it happens in in humans too. We don't know, but the best model to test this is the non-human primate. So the reason that we look at monkeys is because they mimic the multi-organ character of human disease, and because they get the erythema migrans rash, some of them. Maybe I shouldn't tell you this, but it's published. We infected 10 monkeys, and one of them generated a EM rash. But there are other factors, like we use multiple ticks, and I don't know, but interesting. So there's 2 out of 10 are seronegative, and 1 out of 10 develops an EM rash. So they get infection of the nervous system, which mice do not, because it doesn't make sense evolutionarily. You don't want to kill off your host or make them unable to function if you're trying to propagate. And the spirochete burden in tissues is a lot more like humans. Humans don't have a lot of spirochetes, or monkeys don't when they're infected. They're pretty hard to find, but they definitely can cause damage. And so compared to mice, the disease course duration is similar to that of humans. And of course, compared to humans, we can take the tissues from the monkeys and look afterwards and look for infection. And we can do things like lymph node biopsies, which is something that we're going to do. So our hypothesis is that infection with the Lyme disease agent, Borrelia burgdorferi, non-specifically impairs B-cell responses or antibody responses. And so we're going to use 9 monkeys, and this is a project that's in its infancy. It's about to begin. And there'll be 3 groups. So monkeys that are infected, monkeys that are immunized and infected, and monkeys that are immunized only. So really what we want to see is the difference between these 2 groups. So what's the impact of infection on the immunization? And so I work at a primate center, and primates are the best model for HIV. So we have a lot of HIV researchers there, and we don't want to reinvent the wheel. So we know if we immunize with a HIV protein, what the response should look like in monkeys. So that's what we're doing. We're using this protein GP130 from HIV for our antigen, and then we'll infect them by tick bite. And this is what the time course will look like. Infect by tick bite, and at 1 month and 2 months, we're going to give them immunization, and then we're going to boost them, and then we're going to look at their antibody responses over time, and we're going to look at how their immune cells function, both to Borrelia and to the HIV protein. And so that should give us an indication of what effect the Borrelia has on the immune response in general. So I'm kind of short on time, but so that's what we're doing, and this project is funded by Focus on Lyme and the Bill and Marion Cook Foundation. I want to thank them for that. But I also want to tell you about some other things that hopefully will bring you some more hope. We have a diagnostic test that we're developing, and we found that it picks up a high proportion of post-treatment Lyme disease syndrome patients, and I'm excited about that. And we're working a lot on therapeutics. So we have the animal models to test therapeutics. We can take it from an in vitro culture of Borrelia to a mouse model to a monkey model. And so we're working, everything we do is in collaboration. We can't, we don't work in a silo. We have to harness the capabilities of other scientists and put our minds together. And so we're working, we're thinking about biologics. We're trying to determine if there are antibodies that we can synthesize that will target the Borrelia at the same time we treat with antibiotics. We are working with the Duke group, or working with Neil Spector's group. Those are cancer researchers, and they've found a mechanism to identify good targets to kill cancer cells. So we're using the same technology to try to kill Borrelia cells very specifically. We're working with them on that. Ying Zhang, who spoke yesterday, is looking at combination therapies for antibiotic treatment. And, we're testing the combinations that worked well for him in the test tube, we're testing those in the mice. And when we have a good one, then we can take that to the monkey model and test it for efficacy. So, and we're also, anyone who has a new drug or therapeutic that they want to test, we can put it into our animal model system. And I'm optimistic about the future for both diagnosis and treatment of Lyme disease. So with that, I will say thank you to the members of my lab, of course, to my collaborators, and to we have a little bit of federal funding, but we get a lot of support from foundations because they really make a difference in terms of our ability to do translational research for Lyme disease. And so, thank you.